form the three germ layers, so, and you have the axes, so you know that this is gonna be posterior, that end is anterior. You can see where the dorsal mesoderm was, you can see already the ectoderm is changing shape, and that's because the notochord is lying here. The notochord is separating because of convergent extension, it's secreting noggin and cordon, it's inhibiting DMPs, and you're changing the cadherins. So the ectoderm that's going to form mural is going to have N cadherin, the other ectoderm at the side is E cadherin. The notochord is also secreting sonic hedgehog. That sonic hedgehog is going to change the cells that are right above the notochord and form a medial hinge point. Right, and then you're going to start getting folded. And you can see that going on in gastrulation if this is going to work here. Right? So you can see that where the notochord was, you have a change in shape of things. You can see the sides are beginning to fold up. So in frog, gastrulation basically finishes, and then neurulation is occurring all at once in the whole embryo. The anterior of the embryo, the posterior of the embryo. Neurulation, the neural fold is happening along the whole anterior-posterior axis. Right? So now you're about to close the neural tube. You know there's anterior on one side, posterior is on the other side. You can see when you close the neural tube here, it's closing sort of in the middle of the embryo, and then it's going to zip itself up towards the anterior and posterior ends. So when you're forming the neural tube in frog, it kind of forms and closes in the middle, and then it zips up to the anterior and the posterior ends. Right, so it kind of attaches, and then the anterior and the posterior ends zip up. Then when neurulation finishes, you have this neural tube from anterior to posterior that's on the dorsal side of the embryo. Hopefully you saw, and you still can see, the anterior is very different because the brain is in the anterior. There's all these different shape changes going on in the anterior. So a lot more complex things going on in the anterior. Right, hopefully that made sense in frog gastrulation and then neurulation. Chick neurulation is a little bit different, okay? In chick, things are occurring anterior to posterior. So when you look at neural tube closure in chick, it actually starts in the anterior and then it zips itself up going towards the posterior. So again, in the anterior, because the brain is developing there, there's anterior structures that are kind of more complex. So there's strange things happening in the anterior, but you can hopefully see that the neural tube is going to begin to close, and you're going to start zipping it closed from anterior to posterior. If I play this movie here. Right, so you can see it sort of zipping itself up from the anterior, going down towards the posterior. Okay, so chick, which is what this little diagram is trying to explain to you, is that while the processes are the same, you need the same things. You need the notochord to induce neural tissues, except chick does it in a slightly different time sequence than frog does. And that's what this is kind of trying to show you. So this is sort of a progression of chick gastrulation and neurulation and somatogenesis all occurring at once. And hopefully it makes sense in your mind. So if you remember with chick development, you have the posterior marginal zone, you form a primitive streak, the primitive streak moves from posterior to anterior. When it gets to the anterior, you form Henson's node. Henson's node then moves from anterior to posterior. And as it moves from anterior to posterior, it leaves behind it mesoderm, particularly the dorsal mesoderm. So that's what's happened here. You've had development start from the posterior, you've had the primitive streak that went posterior to anterior, you had Henson's node. Henson's node was way at the top. It moved towards posterior. As it did so, it left behind it the notochord. So you see this little red dot in different places. And so the notochord was formed from anterior to posterior. Where's Henson's node now? It's over here, right? So this is Henson's node. So anything anterior to Henson's node has a notochord. But when you look at it, it's sort of a time sequence. Right anterior to Henson's node is where the notochord just formed. So here the notochord is just formed. What's the notochord going to be doing here? It's going to be inhibiting BMPs, it's going to be secreting sonic hedgehog, it's making the neural plate here, and eventually you're going to start forming the neural groove, right? So as you go further towards the anterior, you're kind of going what, back in time, where the Henson's node was. So as you go further back up, you can see you have not only the medial hinge point, but you sort of have dorsolateral hinge points, and you're getting the closing of the neural tube. Right, so because Henson's node moved from anterior to posterior, the anterior represents things that happened a while ago. So in the anterior section, you have closure of the neural tube. In the anterior section, you also have the formation of the somites. If you remember, somatogenesis occurs from anterior to posterior, that's segmentation. So in the anterior, although you can't quite see it in this diagram, you can here, right, you're forming the somites from anterior to posterior. That's what they're trying to show you with these little circles here. As you get closer to Henson's node, you haven't yet formed segmented somites, you haven't yet formed the dorsolateral hinge points. Right, so something like this diagram is easy to test. You're understanding that Henson's node starts from anterior and it moved to posterior. As Henson's node is moving, it's leaving behind the notochord, and with that notochord being left behind, that notochord is going to do a couple things. It's going to induce neurulation, right? It's going to make a neural plate, and then the medial hinge point, and then dorsolateral hinge point, and you're going to get neural tube closure, and that neural tube closure is going to occur from anterior to posterior. That notochord is also going to signal to the paraxial mesoderm, and you're going to start making somites, and those somites are also going to be made from anterior to posterior. Make sense? It's a little different in the frog, because in frog, gastrulation basically finishes and you have a whole notochord, and then you do neurulation. Questions? Yep.
So, I just want to make sure that this is not a mistake. You mentioned that in frogs, that gastrulation occurs from posterior to anterior, right? And then the notochord forms?
Maybe. What do you think? Yeah. Gastrulation occurs from what will be posterior to anterior. So when you start frog gastrulation, this is going to be the posterior. So the mesoderm is starting in posterior and moving to the anterior. So the dorsal organizer moves from posterior to anterior. Once it gets to anterior, it forms a notochord. So now the notochord is formed, it started in posterior, went to anterior, and it's the whole notochord that's going to induce neurulation. And neurulation kind of happens all at once. Interestingly, in frog, it sort of closes in the middle of the neural tube and then zips up to both ends. Our gastrulation looks much more like chick gastrulation, right? Because we form the same sort of flat structure using a primitive streak, etc. Yep.
I think I remember you saying in a different lecture that depending on where cells go through the Henson's node or the primitive streak, that they become like different types of mesoderm.
Maybe. If they go through Henson's node or the dorsal organizer, they are dorsal, just by definition. So the ones that go through that area are going to be dorsal, or vice versa. The ones that are dorsal went through Henson's node. So if you go back to this movie, again, sorry, where's that movie? You know, where this is the dorsal organizer, cells that have beta-catenin and antenna-nodal related and such one. Exactly where are they? It's tough to tell. You can't tell. They will be the ones that form the notochord. So after gastrulation, you can say, all right, these cells were the dorsal organizers. The ones that are sort of to the side of them, they are the paraxial mesoderm. They'll be the next one over. And as you get further over, it's the intermediate mesoderm that's going to form the kidneys. And you get further over, it's the lateral plate mesoderm, right? So exactly where are they? It's impossible to tell unless you use sort of molecular sort of tools and look at it. But the ones that were the dorsal organizer, or the ones that were Henson's node, are the ones that will form the notochord.
Also in the chick.
Also, so Henson's node is in chick, yes. So Henson's node, just by definition, is going to form the notochord. So, you know, when you look at the chick, right, you have a primitive streak that goes one way, and then you form Henson's node. Henson's node is going to move from anterior to posterior, and the, cells involuting are moving, you know, from anterior around back to anterior. There's other cells that are moving laterally, right? So in some parts, there's sort of cells that are kind of close. Exactly where does one end and the other one begin, you can't tell, other than the fact that these will form the notochord, these will not. You can tell after the fact, basically. And that's how you define Hansen's node, because those were the cells that form the notochord. So Hansen's node is chick, dorsal organizer is frog, but they're basically the same thing. They're forming the notochord, the dorsal mesoderm. Okay, and then wherever you have the notochord, you're gonna form the neural tube, and then that neural tube is gonna close. When you've closed that neural tube, though, you've only closed the dorsal aspect of it. It's basically a hollow tube, like a straw. It still has an opening at the anterior side and at the posterior side. Those are called the neuropores. So there's an anterior and a posterior neuropore. You have to close those. If you don't close the neuropores, there's a variety of diseases that you can get. So if you fail to close the posterior neuropore, right, you get a disease called spina bifida, and that's often all kinds of defects in the spinal cord, so the nerves of the spinal cord aren't coming out properly. If you don't close the anterior neuropore, you get anencephaly, which basically means you don't form a brain, and that's often lethal. And you'll see you need to close the neuropores because you need pressure to build up inside. And if you don't close both the anterior and posterior neuropores, you get something that I can't pronounce, and I won't expect you to pronounce it. Just recognize it if it's a true-false question. And I won't attempt spelling for that one because whatever. So if you don't close both neuropores, that's basically an early developmental defect that's lethal, craniorhachypexis or something, which is a complete failure of basically neural formation. To close the neuropores, and indeed to close the neural tube, what you need to close that are several signaling molecules. Well, actually one of them is a transcription factor, PAX3. If you remember, PAX3 is involved in the paraxial mesoderm. So you need the mesoderm around things to help signal for the neural tube to close, whether it's on the dorsal side or the anterior posterior neuropores. You also need sonic hedgehog. Sonic hedgehog also signals to help close the neural tube. You also need folic acid, vitamin B12. When you look at the neural tube, there's receptors, there's folate binding proteins, there are receptors where the neural tube is closing, and you need folic acid for neural tube closure, which is why often pregnant women are prescribed vitamin B12 during pregnancy to help the neural tube close properly. Yep.
So for the diseases, so spina bifida, is it not, like, it doesn't kill the embryo, but if you have anencephaly or the other one, does it make the child not viable?
That's correct. There are cases where you have live births of anencephaly. There's actually pictures in the textbook, I think, where you have almost no head. It's very strange. So it might be born live, but it doesn't really have, it has a bit of a brain stem, but nothing
So
else.
spina bifida is the only one that thinks of viable.
Depends on what you mean viable. So you can get a live birth with anencephaly. It's just,
Like, I mean, like, can you make it through life?
that's correct, yeah, basically. Yep. And there's different gradients of spina bifida as well. Some of them are quite normal. Yep.
So you don't want to break the ancestry?
You don't want, well, just by definition, anencephaly means no cephalic structure, so or very, very little brain. Why, you may ask? Well, that's because you need the closure of the anterior neuropore to cause swelling to form your brain. Okay, your brain is a very, very large structure. And a lot of that size actually comes from internal swelling that causes bulging of the neural tissue, and it's that bulging that kind of allows for further growth. right? So I don't expect you to learn the different parts of the brain, but there's all kinds of different sections of the brain and different folds in the brain, right? Those folds occur because of swelling, right? You close the anterior neural core and there's fluid pressure that builds up inside that causes the anterior part of your nervous system, which will become your brain, to swell. And those swells then turn into the different regions of your brain, so the different, you know, the proencephalon, the mesencephalon, all these other sections of the brain. How do we know that? Because you can actually see that in development, right? That's what this little chick experiment is trying to show you. During development, you form this neural tube. It's a hollow tube and it has openings at both ends. The openings close, so the anterior neural core and the posterior neural core close, and you have this hollow tube, right? That hollow tube is basically where your cerebral spinal fluid is going to go. Your spinal cord is kind of hollow. There's a tube through it, and there's fluid in it. However, during a stage of early development, part of your neural tube actually closes. There's a constriction in your neural tube, basically just below where your brain will form. Okay, so this is my little schematic of things. So this is your neural tube, you just formed it, you've closed the posterior neural core, you've closed the anterior neural core. This is a hollow cavity in here, it's gonna have cerebral spinal fluid in it. If you inject fluid in there, right, you can inject fluid in there. At a certain point in development, in the chicks around day 3, doesn't really matter where, when, the timing doesn't really matter because it varies, but there's gonna be a restriction occurring inside the spinal cord, right? So you've closed that cavity. It used to be open, but it's closed. That cavity being closed allows pressure to build up. Okay, you can see it because if you inject fluid, the fluid doesn't go into the rest of the embryo. But what this allows is for pressure to build up, and that pressure in the anterior, that pressure in the anterior allows the anterior to swell, right? So the brain swells, all the different folds begin to swell. After the brain has swelled, that opening is, that restriction is opened, and then you've opened up the whole channel again. Okay, so it's closed for a little bit, the anterior swells enough for it to start development, and then you open up that channel again. So in you, from your brain ventricles down to the bottom of your spinal cord, you have this continuous opening with the cerebral spinal fluid. But you needed that constriction early in development to cause that swelling. If you had anencephaly, if you had an opening up here, you would never get that swelling because it was open. And if you don't have that swelling, you're basically not going to form any sort of brain structures. That make sense? Yep.
If the pressure creates folds in the brain, then why-
Folds.
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 Yeah, like
Yeah.
folds.
Yeah.
Then why do birds have smooth brains?
You got me on that one. I don't know. Maybe they, I mean, okay, but they still have a swollen brain, even if they don't have folds. Yeah, I did not know that. They have smooth brains?
And koala bears.
Pardon?
Koala bears.
And koala bears as well. Do they not have different regions of the brain? They must have the same sort of mesencephalon,
I
proencephalon.
think they do have regions, but they've discovered to be completely
I-
smooth.
Yeah, I don't know. But certainly in development, because this is chick, and certainly in development, there is swelling and a bit of folding or bulging maybe. Maybe folding is the wrong way. There's distinct bulges, maybe I should say, which are associated with the folds, but perhaps not in chick or koala bear, or probably other things as well. Okay, so this is a little bit of anterior, posterior, changes in the spinal cord, right? So your brain is anterior because you have swelling anterior, etc. So there's a whole bunch of other anterior, posterior specification that's occurring, but we're not going to talk much about that. What we're going to talk a little bit about is dorsal ventral patterning of your spinal cord. Okay, so your spinal cord in your spine is quite large, like it's over a centimeter. It's got, depending on how you count nerves, you know, hundreds of thousands, if not millions of nerves running through your spinal cord. So when you look at your spinal cord, there's actually huge differences between its dorsal side and its ventral side. So what are we looking at? Your spinal cord originated from the neural tube. The neural tube is there because below it is the notochord, and above it is the epidermis, the ectoderm. The notochord has all kinds of things that it's secreting, including sonic hedgehog. The epidermis above it is secreting a bunch of things, including BMPs, and those molecules are responsible for patterning the dorsal ventral axis of the spinal cord. Okay, so again, dorsal ventral means part of the spinal cord that's closer to your back versus closer to your stomach. So it's this 1 centimeter or however big your spinal cord is, but it's still millions of nerves going through here. The nerves that are going through the ventral side are very different than the nerves going from the dorsal side. Okay, make sure you get the orientation right. All of these things are dorsal. Your spinal cord is dorsal, it's on your back. However, the spinal cord is dorsal to the notochord, right? And the epidermis is dorsal to the spinal cord. And your spinal cord has a ventral side, which is close to the notochord, and a dorsal side, which is close to the epidermis. Alright, so the epidermis is secreting BMP. Those BMP signals actually create a structure on the dorsal side of your spinal cord, well, neural tube here, but it will be your spinal cord, and that is called the roof plate. And that roof plate also secretes high levels of BMP. Okay, so the dorsal side of your spinal cord is the roof plate. It's secreting BMP because it's triggered to do so by the epidermis dorsal to it. On the ventral side of the spinal cord, you have the notochord. It secreted high levels of sonic hedgehog, it's still doing so. It creates a structure on the ventral side called the floor plate. The floor plate has high levels of sonic hedgehog as well. Okay, so you have the roof plate on the dorsal side, floor plate on the ventral side. One is secreting sonic hedgehog, the other one is secreting BMP. Therefore, there's a gradient, dorsal ventral gradient, across the neural tube, which will become the spinal cord. And it's within this gradient that the spinal cord puts different types of neurons. There's many types of neurons. I don't know much neurobiology. I know there are sensory neurons and then there's motor neurons, and those make sense in terms of what they do. When you look at where they are, the sensory neurons are going to be on the dorsal side. So sensory neurons run in your spinal cord in areas of high BMP signal, right? So sensory neurons are dorsal. Motor neurons, so the neurons go into your limbs, allow you to move and stuff, are running more ventrally. They're closer to where sonic hedgehog is. Right, so there's different regions of the spinal cord, different types of nerves are placed in different areas, lots of different nerves that you don't need to memorize, but sensory neurons are more dorsal, motor neurons are more ventral. Easy? And you can manipulate this. The reason they are there is because motor neurons are created because of high levels of sonic hedgehog. They're close to the floor plate, which is close to the notochord. If you do experiments where you kind of take another notochord and put another notochord somewhere next to the neural tube when it's forming, it's going to secrete sonic hedgehog. It basically makes another floor plate, right, like region, and then near that you're going to get more motor neurons. So you can create motor neurons in areas where they shouldn't be by increasing sonic hedgehog. Make sense? Okay, so that's sort of neurulation to a large extent. You know, neurulation is totally dependent on the dorsal mesoderm, and then there's some patterning from anterior to posterior, and there's patterning dorsal ventral, but hopefully that's all simplistic and easy to understand. Okay, so now we're gonna kind of continue with something related, which is the neural crest. So the neural crest is actually really complex. There's many, many textbooks just on neural crest. So trying to understand neural crest is very complicated. Yeah, that's okay, we'll start here. Okay, Sometimes it's called the fourth germ layer because it can form lots of different things, right? It's gonna form pigment cells, it can form nerves, it can form cartilage, it can form bone. So neural crest can form lots of different things. If you remember, hopefully this movie works, neurulation is occurring because the notochord, which is here, but it's not drawn in this diagram, is secreting noggin and cordon, etc. It's inhibiting BMP, so the neural plate is gonna have N-cadherin. Far away from it, the epidermis is gonna have E-cadherin, but in between, you have these cells that have a combination of E and N-cadherin. So when neurulation is occurring, and you get this folding, you get the medial hinge point and dorsolateral hinge points, eventually, you have cells that are gonna form the neural tube, cells that are gonna form the epidermis, and these other cells that had a mixture of E and N cadherin are gonna migrate away, they're gonna form the neural crest. And then those cells can migrate to give rise to many different things in the body. How the neural crest work is still hotly examined. There's lots of things that you have to consider with the neural crest, and they're trying to point using this diagram to show you what the neural crest has to go through. So the neural crest basically just underwent the mesenchymal to epithelial transition. Sorry, epithelial to mesenchymal transition, right? They're mesenchymal cells now, they're gonna migrate away. But they need things, and these are things that we're gonna consider and talk about the next bunch of slides. They need some sort of long distance guidance cues. They're going towards something. Something is attracting them. And often we don't know what is guiding neural crest cells. Because neural crest cells undergo really complicated migration paths, right? But they're going towards something. They have to move on something. They have to move on some sort of substrate, right? We already know cells are gonna move on ECM using integrins, so they need the right ECM and the right integrins to move. Okay? There's a lot of different ways you can guide them. So you can allow them to move or not allow them to move. You can stop them and start them. You can also put up barriers, right? So there are physical barriers present that won't allow them to go certain places. There's lots of things, lots of ways that neural crest cells can be guided to go to the right location. Neural crest cells, unlike the paraxial mesoderm, if you remember the somites have information from the Hox genes, right? So the retinoic acid going from anterior to posterior, and the somites have different Hox genes. So if you transplant somites, they already know their position. They know their anterior, posterior position. Neural crest cells do not know that. Neural crest cells don't really get faded until they get to where they're going. So as the cells are moving, they're picking up positional information. But they don't necessarily know their fate until they get to where they're going. For them to move, though, they have to do a couple of things. Okay, so part of the process of EMT is that they have to, number one, is they have to break their cadherin links, right? So again, in the neural tube, you're gonna have N-cadherin. In the epidermis, you're gonna have E-cadherin. You do not want these neural crest cells to have cadherins. You don't want those cadherins to work because you don't want them to stick to anything. You want them to be migratory. So they express a protein called SNAIL. So SNAIL breaks cell-cell junctions. SNAIL breaks cadherins basically. It doesn't allow cadherins to function. So neural crest cells express SNAIL. There's also a similar protein called SLUG, which is almost the exact same thing. And the reason they're called SNAIL or SLUG is if you mutate them, if you knock them out, the cells move much slower. Why do they move slower? Because they are making cadherin junctions. Right? So you need SNAIL or SLUG to be functioning so you can break the cadherins to allow the cells to move. The other thing you need for them to migrate is they need to activate their actin cytoskeleton to allow migration, and to do that you need Rho GTPases. So the neural crest cells are going to be expressing SNAIL to break junctions and Rho GTPases to allow them to alter the cytoskeleton to allow them to move. And if you do experiments the appropriate sort of manipulations will cause them not to move, right? So if you change their cadherin expression, if you increase cadherins, potentially they're not gonna move properly. If you decrease row proteins, or if you decrease slug or snail, they're not gonna move properly. Additionally though, if you change noggin or BMP, the reason these cells don't have cadherin, don't have a specific level of E-cadherin or N-cadherin is because of the level of noggin and BMP. Right, if you start interfering with noggin or BMP levels, you're gonna interfere with the level of snail, interfere with the level of Rho GTPases, and these cells won't migrate. Alright, so the neural crest originate as the neural tube closes. Okay, as the neural tube is closing. Things vary quite a little bit. So different animals do things slightly differently. In some animals, neural crest actually migrates before the neural tube closes. But in general, the neural crest migrates when the neural tube closes. So as you get neurulation occurring from in the embryo, in the chick it occurs from anterior to posterior, right? You have cells that are going to be moving away from the dorsal side, these neural crest cells, from anterior to posterior in the embryo. And there's different regions of these neural crests that we'll talk about individually. So there's cranial neural crest, as it suggests, that happens in the head. Head structures are very difficult to study because the brain dominates things. So the head is often very big and it's very difficult to figure out what's going on, but we'll talk a little bit about cranial neural crest cells. Cardiac neural crest cells, that's pretty self-explanatory, they're going to contribute to the heart. There's something called vagal and sacral neural crest cells that are sort of two regions of the embryo. Sort of sacral is down at the posterior and vagal is sort of further up. That they're involved in sort of nerve formation, but we're not really going to talk about those guys. What is most well studied are something called trunk neural crest cells. So the trunk is almost everything from your neck down to your butt. And those are the easiest one to study. Okay, so most studies are on trunk neural crest cells, and then what we learn from trunk neural crest cells, we try to apply to the rest of the neural crest, the rest of the cells, neural crest cells. Okay, so again, there's a lot of cells moving around. So this is sort of a fluorescent image of the cells moving in different places, and you can see there's many cells that are moving. There's many different parts of the embryo. Most of what we know from about neural crest is from the trunk. So let's look at trunk neural crest cells first, and then we'll look at cranial and cardiac a little bit after that. Okay, so trunk neural crest cells. So trunk neural crest cells originate on the dorsal side of the neural tube right when you have closure of the neural tube. Okay, so in this image you're looking at the neural tube in the middle, nicely fluorescent green. You can see the notochord below it. So you can see the neural tube, and above it is the epidermis. So the neural crest cells were on the dorsal side of the neural tube. In the trunk, these neural crest cells are going to migrate away. They're going to migrate away from this dorsal side. When they migrate away, they take two different routes. They take a ventral route or a dorsolateral route. Okay, so we'll look at this route in 4 different ways. So this is one way to do it, but we'll do it several times. Okay, so again, maybe I should back up a little bit. Let's go to this movie again. Okay, whoops. So, you have these neural crest cells. They're going to form on the dorsal side of the neural tube, and then they're going to migrate away. Okay, where are they going to migrate? There's different routes that they can take. One of them you'll see is ventral, so one of them is going to go sort of towards the ventral side, and one of them is dorsolateral, so they're kind of going to go dorsally and away. Okay? So, one of them is ventral, so they're going to go down next to the neural tube through the somites moving ventrally, and the other one is sort of dorsolaterally, where they're going to be moving just below the epidermis through the dermamytome, or above the dermamytome, and you'll see that in a second. Okay, so this is one representation, hopefully not the easiest to understand. This is another little diagram from the textbook, which may be equally confusing, but let's go through this, and hopefully, things will make sense. All right, what are we looking at here? Rostral caudal, I don't know why they do that. So this is anterior, posterior. Okay, so this is the anterior half. All right, you can see the neural tube, good, that makes sense. You can see the notochord, that's good too. On either side of the neural tube and the notochord is the somite. Okay, the somite is made up of different components. Here they've chosen to separate it for whatever reason. So you have the dermamyotome and the sclerotome. So this whole thing is the somite, except they sort of separate the dermamyotome from the sclerotome. The somites are segmented, right? They go from anterior to posterior. So you can see the segments of the somites here, okay? Each somite has an anterior and a posterior half. We already know that because the anterior half is going to have the efferent, and the posterior half is going to have the efferent receptor. I may have that backwards, but it's down here actually. So anyway, we'll get to that in a second. Don't worry about that. All right. So on the dorsal side of the neural tube, you have the somite. These are trunk. On the dorsal side, you have the neural crest cells. These are trunk neural crest cells. So the trunk neural crest cells can take two routes. One of them is ventral. That ventral route goes through the sclerotome of the somite. Okay, the ventral route goes through the sclerotome of the somite. But it only goes through the anterior half of the sclerotome of the somite. Why does it only go through the anterior half of the sclerotome? Well, a couple of reasons. One is it needs something permissive to migrate on. So for example, there are permissive molecules in the anterior half but not on the posterior half, things such as fibronectin. So these neural crest cells can migrate through the anterior half of the sclerotome because they can use their integrins to migrate to things. Those permissive molecules are not present on the posterior half, so they can't migrate. Okay? Additionally, there's inhibitory molecules in the posterior half. Okay? So in the posterior half is efferent. Right? If you remember, efferent is on one half, the efferent receptor is on the other half. That is involved in segmentation of the somite, but those molecules are still there and they can be used again. In this case, efferent is on the posterior half, and the receptor is on the neural crest cell. Okay, so now the neural crest cells are repulsed by the posterior half of the sclerotome. So not only does the posterior half of the sclerotome not have fibronectin, but it actually has efferent, the ligand that's going to cause them to repel it. We'll see that again in a couple of other slides. In addition to all that, there's another molecule called semaphorin. A lot of these other things are coded by semaphorin. For example, you have the neural tube here, right? Why don't the neural crest cells move into the neural tube? Well, because there's other molecules between things called semaphorin that are just repelling things, just acting like a physical barrier. Okay, so that's the ventral route. The ventral route is going through the anterior part of the sclerotome because there's fibronectin there, and not the posterior part of the sclerotome because there's efferent there, and the efferent receptor is on the neural crest cells. Okay, so that's a little bit complicated compared to the dorsolateral pathway. Some neural crest cells move dorsolaterally. They just move underneath the epidermis above the dermamyotome. So dorsolateral just goes along the entire back of the embryo, but just underneath the epidermis. And you'll see other pictures of this in a second. All right, so that's one way to look at it. Here's another way to look at it. Okay, you have somites. There's one somite, two somites. This is the anterior half of the somite, this is the posterior half of the somite, anterior, posterior of the somite. When you look at where ephrin is, if you remember ephrin is the ligand, ephrin is on the posterior half of the somites. When you look at where are neural crest cells migrating, neural crest cells are migrating on the anterior half of the somite. Why? Because ephrin is on the posterior half. Another way to look at it, anterior, posterior, you have the neural tube here. You have somites next to the neural tube. Each somite has an anterior and a posterior half. Neural crest cells are going to migrate through the somites. They're going to go ventrally through the somites, but they can only migrate through the anterior half of the somite. Why? Because the anterior half of the somite has permissive molecules such as fibronectin, and as importantly, the posterior half has ephrin, which is repulsive. Make sense? When these neural crest cells are migrating through the somites, those somites have positional information. Each somite is different because it has different Hox genes. So the neural crest cells that go through anterior somites are going to get different signals and therefore have different states in terms of what they're going to be. They're getting positional information by going through the different somites. They're picking up sort of cues as to what they're going to differentiate into eventually. Not only do the neural crest cells migrate through the anterior half of the somite to pick up information, however, but in addition, motor neurons, if you remember, motor neurons come out from the ventral side of the neural tube. Motor neurons are going to be going out and innervating different things. Motor neurons are also passing through the anterior half of the somites. Those motor neurons, when they pass through the anterior half of the somites, are also picking up information. They will know where they're going to go and what they're going to become. Okay, so somatogenesis and forming those somites that have different positional information is not only important in forming different types of muscle, but those somites are then also used to give positional information to things such as neural crest cells and motor neurons that pass through them. Cool? Okay, another way to look at it. Alright, so again, this little diagram will be on your final exam. You should be able to recognize things. There's a neural tube here, there's a notochord here, paraxial mesoderm, intermediate mesoderm that's going to form the kidney and the gonad, and lateral plate mesoderm. Here are the neural crest cells. They're going to form on the dorsal side as the neural tube forms. Right? You should be able to recognize things, like I said, that NT's neural tube for my little thing. The somites are going to the neural crest cells, the trunk neural crest cells are going to take one of two routes. One of them is going to be ventral, that's going to go through the somite, and the other one is dorsolateral, it's going to go in between the somite and the epidermis. Right? That ventral path is going through the sclerotome, and the other path is going next to the dermomyotome. If you want to look at it differently, so I've switched the axes just to kind of mess with your minds. This is anterior, this is posterior. Okay? You have the neural crest cells here. These are the trunk neural crest cells. They're going to migrate. Some of them are going to migrate dorsolaterally. Dorsolaterally is just under the epidermis on top of the dermomyotomes. So the dorsolateral migrating cells are going to migrate basically over everything. They're using to migrate over all of the somites. Dorsolateral neural crest cells are going to give rise to several things. One of the things they're going to give rise to is melanocytes. So all the pigment stuff came from neural crest cells that migrated dorsolaterally. Okay, and things are quite conserved. The migration routes are quite conserved, and the genes that regulate things are pretty conserved. It's sort of interesting when you start mutating some of the migration genes for melanocytes, the same gene defect is going to have the same effect in sort of conserved across mammals. So you can see this one with this mouse has the same sort of pigment deformation. But the melanocytes are what gives you the stripes or patterns or skin color or whatever, hair color, etc. Okay, so that's dorsolateral, moving across the entire surface, whereas the ventral is only going through the anterior half of the somite, through the sclerotome. Okay, why? Because there's effort in the posterior half. These anterior half migrating neural crest cells are going to give rise to several things. One of the things it's going to give rise to is sympathetic ganglia, so basically your fight or flight autonomic nervous signals. Okay, hopefully this explains things okay. We'll start with this again next lecture. All right, see you guys on Wednesday.